Okay, now on to new stuff. Molecular homology. Looking at similarities in especially the DNA, but proteins and other molecules as well are important here also. Now, of course, Darwin didn't know about DNA. It wasn't until the mid-1900s that people began to develop the technology needed to generate sequences for proteins or DNA. Once they did, it was discovered that different proteins have distinctive features and that there are differences and similarities we can look at in different organisms. Over the past few decades, there have been quite dramatic technological advances in this regard. Back in the 70s was the very first DNA sequencing. 50s was protein sequencing. By the 80s, we had the first techniques to make lots of copies of particular DNA in order to be able to study it. And today, there are increasing technologies to sequence large amounts of DNA from particular organisms. Once we have that sort of data, we can start comparing how similar or different is the DNA, are the proteins, things like that, between different types of organisms. And it turns out that these patterns of similarities are a good match for the evolutionary relationships already inferred based on looking at things like anatomy and other more large-scale similarities. Now, yes, it's not a perfect match. For one thing, a lot of things had not been studied very carefully. So you had stuff that, well, yeah, that kind of looks like that. We'll just put it in that category for now. Maybe someone will study it later. And then coming along with the DNA, you didn't really look closely at this. These come out different on the DNA, and yeah, when you look at them, they are different. So there are plenty of examples of that sort of thing where the DNA analysis leads us to go back and take another look and revise things. But overall, when you look at the DNA data, when you look at the morphology, it all fits together, pointing to similar patterns. There are also cases where the analysis was not so good. But again, as we study more, we work these out and figure out those issues. So we get very good patterns of genetic similarities supporting the idea of evolutionary relationships being shown by these molecular features. In addition to the DNA, the protein coded in them, you do have things like, say, does an organism use a particular chemical? For example, cholesterol is a lipid that is made by animals, used in our cell membranes and things like that, not in other organisms, tying into the relationship between animals. Bacteria, you may hear mention of gram-negative versus gram-positive bacteria. Gram-positive bacteria are ones that stain with gram stain. Gram stain is a stain discovered by Gram, which happens to bind to a particular molecule found in the cell walls of some bacteria, which are related to each other. They share that molecule, and their DNA, other lines of evidence, supports them being related. We even see, at a very basic level, all living things, bacteria, archaea, eukaryotes, have some similarities in our DNA, in proteins, things like that, which support the idea that ultimately all life on Earth has a common ancestor evolutionarily. For example, basic use of DNA, same four bases, same four bases in the RNA, same 20 amino acids in protein, and how you use the three of them. DNA as the information storage, gets transcribed into RNA, translated into the protein, using the same approach with our tRNAs and messenger RNA and ribosomes, all that. Fits well with the idea that all of these are tracing back to a common ancestor that had this basic chemical setup that we see continued into all modern life. Here's an example of a specific comparison. This is in the non-coding region, so that would be things like introns and spacer regions, things like that. In beta globin gene, so that's one of the genes helping make hemoglobin, oxygen carrying in the blood. And this is how different it is from human to these other things. We're most similar to the chimpanzees, only a little bit more different from gorilla than orangutans, gibbons, old world monkeys, new world monkeys, other primates in there. And now this is just one little bit of the DNA, but when we look at much larger scale, whole genomes now we can get, and we see the same basic pattern that humans are most similar in our DNA to the chimps, a little bit less to the gorilla, and so on out for these groups. One complication, there are several different ways you can look at and compare DNA. So depending on how the calculation was made, you can see different numbers of 95% similarity or 98% similarity or all sorts of different variations there. The exact number, okay, however you want to do your numbers, but this pair of similarity. And we can do the same sort of thing. I'm going to say particular type of clam. Okay, here what's most similar to it. Ones that are a little more different, so on. Across the board, whatever type of organism you want to look at, you can do these comparisons and get a pattern fitting well with what we expect evolutionarily. Ontogeny. Okay, well, what is it, first of all? Ontogeny is the series of changes that takes place during the development of an organism. So, for example, we as humans start out as a fertilized egg, zygote, just like any other animal, or for that matter, practically any eukaryote. And then that starts dividing. We get a little hollow ball of cells, a blastula, folds in, gastrulation, and layers develops into a more recognizable embryo. Birth, you have a baby with noticeably different proportions than an adult, and growing through lifespan. So those are ontogenic changes in humans. Some things, such as these, have rather more drastic difference between a larval stage and the adult. And when we look at ontogeny, we see a couple things. One, early in development, different types of organisms often look more similar than the adults do. And so it's hinting a bit at a relationship not as obvious in the adult. Here's a classic example. The acorn worm here and the starfish do not look much like each other. But when we look at the larvae, both of them have a rather complexly squiggly band of cilia around them. There are some similarities in the build of these two larvae. And in fact, when we look at the DNA, we likewise see that the hemichordates, the phylum acorn worms, and the echinoderms, the phylum of starfish, are closely related to each other. Another thing we see in ontogeny, sometimes there's a feature that we can see in a stage of development that looks more like the ancestor than like the modern form. And so we see connections of that sort as well. Sometimes there are things that appear earlier in development that aren't particularly useful anymore to that organism, but would have functioned in the ancestor. For example, human embryo, rather early in development, has a tail and has gill slits in the neck region. Tails aren't particularly useful to us as adults. Living on land, we don't need gills. And so these change and basically disappear or change into other things through development. But the presence of these early on development fits in with having had ancestors that had tails and had gills. Classic studies on this go back to the late 1800s to early 1900s with work by folks like Ernst Haeckel, a German embryologist. He was skilled artistically, made good drawings, not just of embryos, but of a number of other things. And did a lot of work on this. And his pictures got widely copied. But sometimes they weren't completely accurate. Sometimes he exaggerated a bit to match his ideas on evolution and development. Now, of course, this could partly be a matter of trying to look through the microscope, and this is not something easy to do. First of all, you have to actually find the specimen at the right stage of development and observe it microscopically. And microscope technology was not as good back then as it is now. So this is difficult work, and Haeckel probably at some points kind of peering in the microscope. I know there should be something there. I think I see something. He may have been misled that way, or could also be some examples of kind of, okay, that doesn't look so great, but and I know what it should be like. I'll make a drawing that shows it better to be a nicer picture. So this isn't deliberately I'm going to try to fool people, but there are some errors in his drawings. But correcting them fits better with a modern evolutionary understanding. Haeckel's big idea was that ontogeny was basically a museum of evolutionary history, and new features kept on getting added on to the end. So as you go through evolution, you'll be adding new development beyond what happened previously. A couple of big problems with that. One, it doesn't always happen that way. There are a number of examples of things where actually going back to a previous stage is what leads to further evolutionary development. For example, in humans, if you look at pictures of baby apes, they tend to look somewhat more cuter than their parents. Two reasons. One, babies in general have big eyes and ears, things that we associate with being cute. But in particular, the baby apes also look somewhat more human than their parents do. One part of that is that their brain and head is larger compared to the body than as they grow up. Apes stop growing the brain relatively early. I mean, they got good brains for animals, but not real impressive compared to us. We keep growing the brains till about 5 years old or so, not just right around birth. And so we're growing our brains more like a baby ape than a young ape. we're kind of retaining a baby-like feature on later in life than them. By Haeckel's reasoning, that means that the apes are more advanced and more primitive in our pattern of development. No. They've got somebody who works for them, we got somebody who works for us. But it illustrates you can go back. You're not just adding on new things to the end of development. You can go back and turn off another way from an earlier point in development as well. Another thing about Haeckel's idea, ontogeny can't just be a museum. All stages in life have to survive. And so they will be subject to evolutionary pressures, can change in various ways, things like that. They're not just going to preserve exactly how things were in the past. So we should see variation between embryos of different types of things. It's not going to be perfectly identical, but there are similarities pointing to evolutionary relationships. Here's an example. We have fish, chicken, and human embryos. And early on, they look more similar. Now, you get confusion. No, this does not mean that you were a fish when you were small. It means that vertebrates in general start out looking more generically, okay, this is an early developing vertebrate. And then as they develop further, they begin to get more distinctive features that make it more obvious what kind they're going to be as they grow up. So we do have some key similarities, but differences as well. For example, in the DNA, the patches shown in kind of a red-orange color here are areas where a particular key gene turns on, same basic gene in all of them, that says start growing this into an appendage. Now in the fish, you've got other genes that say make it a fin. In the chicken, the wing, and us, an arm. But it's the same basic grow a limb here gene. So we have basic similarities in a lot of the key genes regulating the processes. We have relatively similar looking features early on, and then as we grow, become much more distinctive.
Is that
Is that a good point to pause?